For more than 40 years, there's been one technique that's been the absolute gold standard for reading DNA. It's the method that first cracked open the book of life, and it's called Sanger sequencing. Today, we're going to break down the simple yet totally brilliant trick that made it all possible.
Okay, so imagine you've got this book, right? It's written in a language with only 4 letters, A, T, C, and G. But it has billions and billions of words. That's pretty much the challenge of genomics. For the longest time, the genetic code was a complete mystery. We knew the letters existed, but we just had no way to read the actual sequence. So how on earth did we finally figure it out?
While the big breakthrough came from a truly brilliant English biochemist, Frederick Sanger. He came up with a method so clever and so elegant they called it the chain termination method. It was this amazing chemical trick that could literally stop the DNA copying process at very specific points. This allowed us, for the first time, to read the sequence, one letter at a time.
So, how did he do it? How did Sanger figure out how to hit the pause button on the biological machinery of life itself? It wasn't with some crazy complicated machine. It was all thanks to one single, very special little molecule that acts like a stop sign for DNA. Meet the secret ingredient, the dideoxy nucleotide, or what scientists call a DDNTP. Now think of a regular nucleotide like a Lego brick. It's got connectors on both sides so you can keep building the chain. A DDNTP, though, is a special brick. It's missing the connector on one side. See, a DNA chain normally grows by adding new bricks to a spot called the 3' hydroxyl group, but a DDNTP doesn't have one of those, so the second it gets added to the chain, well, that's it. The chain has nowhere else to grow. It is literally terminated.
So, how do we actually use this stop sign in the lab? It's a fantastic bit of chemistry. You take a test tube and you mix your DNA template, a primer to get everything started, an enzyme called DNA polymerase, and a ton of regular nucleotides. But, and here's the key, you also add a tiny little bit of those colorful DDNTP stop signs. As the polymerase starts copying the DNA, it's mostly grabbing the regular building blocks, but every now and then, just by pure chance, it grabs a color DDNTP instead. And bam! The chain stops right there. This process happens millions of times, creating a massive collection of DNA fragments of every single possible length, and each one is tagged with a color that tells us exactly which base was the last one to be added.
Sanger's chemistry was an absolute game changer, but the original version was incredibly slow and a ton of work. So how did we go from painstakingly reading one gene to sequencing entire genomes? Well, the answer is a full-blown technological revolution.
And the leap forward was just immense. I mean, look at this. On the left, you've got the original method. You had to run 4 separate reactions, one for each letter, using radioactive labels. Then you'd run them on these giant, clunky gels and read the sequence off an X-ray film. It was brilliant, for sure, but so slow. Now, look at the right. That's the modern way. Thanks to fluorescent dyes, with a different color for each DDNTP, everything happens in one single tube. No radioactivity, no 4 separate lanes, just one clean, automated process.
And this timeline just shows you the incredible speed of innovation. From Sanger's discovery in '77, it took less than a decade to get automated sequencers using those fluorescent dyes. By the '90s, things got even faster with capillary electrophoresis, which let machines run 96 samples at a time. This high-throughput power is what made the impossible possible, sequencing the entire human genome. The first draft was published in 2001, a truly monumental achievement built right on the shoulders of Sanger's original idea.
So we've got millions of these colorful, chain-terminated DNA fragments. But how does a machine turn that jumble into the A's, T's, C's, and G's that we can actually read and understand?
This is what it spits out, a chromatogram. The machine separates all those fragments by size in a tiny little glass tube, with the smallest one zipping through first. As each fragment passes a laser, its colorful tag lights up. The machine records the color of each peak. One color for A, another for C, and so on. And the order of those peaks gives you the exact DNA sequence. It's basically like reading a genetic barcode.
But hold on. A raw sequence like that is just the starting point. To be useful for science, or especially for medicine, that sequence has to be ridiculously accurate. And that means there's a serious quality control process that has to happen next. So, scientists don't just take the machine's word for it. The data gets cleaned up, but the most important check is this.
They sequence the DNA from both directions. It's kind of like reading a sentence forwards and then reading it again backwards just to make sure you got every single letter right. By comparing these two separate reads, they can catch and fix pretty much any error, making sure the final sequence is incredibly accurate. Now, we live in this age of what's called next-generation sequencing, which can read a whole genome in a single day. So you might think Sanger's method is kind of a dinosaur, right? Obsolete? Well, you'd be wrong. It still holds a really unique and absolutely critical role in science. And the reason really comes down to a trade-off. NGS is amazing for discovery, for sequencing millions of things at once to just see what's out there. But its reads are short and it can have a higher error rate. Sanger sequencing is the total opposite. It's slow and steady, it focuses on just one piece of DNA, but it produces these beautiful, long, super accurate reads. It's like NGS is the scout that explores the whole territory, but Sanger is the expert surveyor you call in to confirm exactly where the treasure is buried. And this number right here is exactly why. The accuracy of a well-done Sanger sequence is 99.99%. That is an astonishing level of precision, and it's why it is still the undisputed gold standard for validating genetic findings. When a scientist uses NGS to find a mutation that might cause a disease, they use Sanger sequencing to prove, without a doubt, that it's really there. And hey, that's not just a cute nickname. It's a scientific fact. The whole community refers to it this way. Just look at this quote from a scientific journal. Sanger DNA sequencing has served as the gold standard. It is the benchmark. It is the final word when you need to be absolutely sure about a sequence.
So it all leaves us with this really interesting question. In a field that changes at lightning speed, Sanger's method has stuck around for nearly half a century. It's a testament to the power of a simple, elegant idea, but it also makes you think, what's the difference between a technology that's just a stepping stone and one that becomes a cornerstone of science? What really makes a discovery timeless? Is it precision, reliability, or just pure, simple genius?